have this, and I will be. All right, so today I'm going to be focusing specifically on the coronavirus and how it goes about replicating and hijacking our host cell machinery. It's going to be a bit of a shorter lecture, I would say, hopefully, but it is a very dense lecture. I will try to highlight some of the stuff that I do want you guys to know for the exam. And if you guys have any questions about it, feel free to ask at the end or throw it in the chat. So the general outline of this presentation is going to be, basic intro to the different kinds of coronaviruses that exist, and then some of the key structures that are within the actual viral particle, and then highlighting key receptors that the coronavirus uses to enter a cell. And then once the cell actually, or the virus deposits its genomic material into the cell, what the replication process is for that. And then I'm going to end it with relating this to health and disease and talking a little bit about why certain groups of people are more susceptible than others. So I didn't delete that, but what I want you to know are some of the key structures of the virus. So this is both structural and non-structural proteins, which I'll get into depth in a few slides. I want you to know the receptors that facilitate the coronavirus entry into the cell. And I also want you guys to know the coronavirus replication process. It's kind of complicated, if I'm being honest, but this will be the only virus that infects humans. We're going to talk about bacteriophages tomorrow, that I want you to know the actual life cycle for. Bacteriophages will be something separate tomorrow, and I would like you guys to know that as well. But I'm going to keep it, for the most part, pretty simple. And then I want you to understand why patients succumb to the infection, or at least those groups that are more susceptible. So there are 7 known coronaviruses that infect humans. And so it's thought that the common ancestor with coronaviruses began with the bat. And basically, there's a variety of different coronaviruses that circulate within bat populations. But what ends up happening sometimes is this thing called species jumping or genetic shifting, where there is some kind of mutation within the virus and it's able to infect another species or host. And so there's 4 different genuses of coronavirus, so alpha, beta, gamma, and delta. But the only ones that are able to infect humans are the alpha and beta coronaviruses. And so typically, coronaviruses before the pandemic are associated with the common cold. They're responsible for, I believe, up to 30% of the common cold. I think rhinoviruses and one other virus are the 3 that make up why you get colds. So the ones that I have highlighted here in purple are the ones that typically circulate within the human population, and they usually don't cause any severe disease. It's just usually, it stays within the sinuses and doesn't actually make it all the way to the lungs. Now with SARS, where we had an outbreak in 2003, MERS, and now the COVID-19, those 3 are considered ones that basically jump from species to species. And when that happens, the virus tends to be virulent, or it tends to be really effective at circulating amongst populations, and it tends to create severe disease. And it also has to do with where these 3 viruses are able to actually penetrate within our system. So as I mentioned, with these ones highlighted in purple, they stay mainly in the sinuses. However, with these 3 other viruses that cause more severe disease, they're able to make it into the respiratory system. Once it reaches that, that's when severe disease can manifest. So I really want you guys to know, I'm going to be focused mainly on these 3 because those are the ones that really create problems for us. And there are going to be some parallels between the 3, but I'm going to be focused mainly on COVID-19. So the three that I just talked about are the ones that jump from species to species. And so again, bats are thought to be the main host that allow the virus to jump from other species. And it's hypothesized because of this, because there's so many different coronaviruses that exist within the bat population. Within China, it's thought that there's over 500 different COVID virus variants. It's thought that from jumping from species to species, it's picked up different mutations that allow it to manifest in the severity of diseases. So basically the SARS virus that came out in 2003, it's thought that it came from bats and it jumped to civet cats or other small carnivores, and then it ended up spilling over into humans. Whereas, and then this epidemic ended, I forget how long it actually lasted for, but it was a short amount of time. There's only 8,000 cases and only a few deaths compared to, COVID-19. Now, with the MERS virus, basically it also originated from bats, but it ended up transmitting to camels. And this is the Middle Eastern respiratory syndrome. And so, they in the Middle East, they still use camels quite frequently. And this virus still circulates amongst the camels. And because of the mutations that are able to occur within the camels, infections are still occurring because this virus is still able to species jump into humans. So why is this pandemic harder to control than the previous 2003 SARS? Well, it has to do with knowing what the the spillover reservoir is. So it was back in 2003, we were able to identify quite quickly that the main reservoir was civet cats. I'm not really sure if they ended up killing a bunch of them to stop the spread or what exactly. But I think knowing where the source is, you're able to go ahead and break this chain from causing the virus to spread globally. Compared to the COVID-19 virus, at the time of the virus, when the pandemic began, we weren't really sure what the source was. We had an idea that bats were going to be the main cause, but we knew that it ended up species jumping into something else, and it's thought that these pangolins are the source of this. With the 2003 SARS virus, the transmissions typically happened within a hospital setting. So it's usually pretty contained and wasn't able to spread compared to the current pandemic, where basically a lot of it was found in public settings. And this public settings basically cause an exponential rate of infection. The other thing that also contributed to reducing and controlling the 2003 epidemic was the transmission didn't occur until about 2 to 3 days after symptoms began. And most of the time, you were only able to transmit the virus if you experienced symptoms. There's very low cases of people that were asymptomatic that were able to spread. Compared to COVID-19, there are lots of people that are asymptomatic and they have the ability to spread the disease. And being asymptomatic and not really being aware that you have it, and you go out into the public, especially people that aren't wearing their mask, make it a lot easier for the virus to spread. And because it's all occurring in the public, we have the pandemic that we do. And luckily, at least in the United States, it's calming down. So the majority of the lecture is going to be focused on viral replications. And here is a basic diagram of how viruses in general end up getting into a cell. So first, they end up binding to some kind of cell receptor. And every virus has its own specific receptor or set of receptors that it needs to get into a host cell. And so I'll be talking a lot about those receptors that are implicated in COVID-19. Once the virus is into the cell, there's some kind of process where the coat ends up being taken off of the virus. And the whole point of this is so the genetic material, whatever the genome of the virus, so if it's a DNA or RNA virus, it's able to get deposited into the cell so it can go ahead and hijack host cell machinery. And so typically what's going to end up happening is the virus is going to be made up of a couple of different genes that are going to encode for a specific amount of proteins. And these proteins generally are going to be encoding for non-structural and structural proteins. And you'll hear me talk more in depth about this, and I'll reiterate a couple of these points just to drive these points home. With the structural proteins, they're basically going to be implicated in actually creating the structure of the virion. And those are usually going to be expressed later on. Versus the earlier genes, those are going to be segments of the genome that encode non-structural proteins. And what these proteins usually do are there's some kind of enzyme that play a role in viral replication. And then, so once all of these steps are completed, the virus is able to assemble again by hijacking host cell machinery, and then it's able to bud out of the actual cell and basically repeat the process by infecting other host cells until the virus ends up making you sick due to the immune response. And it's not until the immune response really gets a handle on the virus that you're able to clear these viruses and prevent this process from continuing. So what I really want you to know are these key points of the coronavirus. So I highlighted some of these in the intro to virology, but these are the things that I have highlighted here, things that I really want you to understand and know about the viral structure. So I do want you to know that the coronaviruses have a positive sense RNA genome. And this is going to be protected by a nucleocapsid as seen here. On the surface of coronavirus, it's going to have a couple of key structural proteins. One of them is going to be the envelope. And so the envelope proteins are usually consistent of both viral proteins as well as host proteins when it's budding out of the cell after it completes the whole replication process. The other really important protein that I'm going to emphasize a lot, not only in this lecture, but also when we're talking about vaccines against the virus, because this is what our vaccines are going to be against, is the S or spike proteins. And these proteins are essential for the virus to get into a host cell. If it doesn't have these spike proteins, or if it doesn't have the receptor that it needs to bind, the virus will not be able to get in and infect you. The other protein that I want you to be aware of are these membrane proteins. And these are going to be really the most abundant proteins that make up the coat of the virus structure. And this is going to connect basically the envelope and the nucleocapsid together. So here I have an image of with a close-up of the spike proteins. And so again, these spike proteins are going to facilitate the entry into the host cell. And there's going to be two important areas of this that I want you to know. There's going to be this ACE2 binding domain. So that's going to be where the virus actually binds to the host cell receptor. And then there's going to be this lower part that's called the fusion portion. And so this is going to be the part that actually allows the virus to fuse with the host cell membrane and deposit its genetic material into the cell. So the ACE2 binding domain, so as seen here, will end up binding to the ACE2 or angiotensin converting receptor enzyme. And this is expressed by specific host cells, which I will get into a couple of slides. The fusion portion, again, is going to be how the virus is able to fuse with the host cell. What I really want you to pay attention to here, though, is what's conserved and what's variable within this this spike protein. So basically what scientists were able to do is take all the different kinds of coronavirus variants for the specific virus, and we're able to basically superimpose all of these together. And they wanted to see, what do all of these viruses have in common and what is difference amongst them. And so it seems like what they have most in common is this fusion peptide. So it seems to be the area that's most conserved. As opposed to the binding domain for the ACE2 domain, this area seems to be more variable. And this might play a role in pathogenesis as making the virus more or less efficient. So depending on what amino acids or proteins are expressed in this area might indicate how efficient the spike proteins can actually interact with this ACE2 receptor. And it might play a role as to why this virus is so, it can easily spread or easily get into cells compared to other non-virulent viruses. So here I have a diagram of the actual facilitation of the binding and fusion of the host cell. So basically, as I mentioned already, and I'm going to keep on emphasizing, the spike proteins are going to bind to this ACE2 receptor. And so what I'm really going to do is highlight this interaction that's going on here, and then I'm going to go a little bit in depth here. You don't need to know this. I find it to be interesting just because I like structures of molecules, if you haven't been able to tell from all my lectures. But it's interesting to see how, proteins are able to fold depending on what happens to them. And that helps with the facilitation of this whole process. So once the ACE2 receptor binds to the, or actually the other way around, the spike proteins bind to the ACE2 receptor, you basically are going to have this protease that's called TMPRSS2. And so protease is just something that, it's a type of enzyme, note the ACE. And depending on the protease, it's either going to break apart the protein or it's going to degrade it completely. Somebody asked, is our plasma membrane unable to identify that what's entering in is a cell? Not exactly. So it's going to be your immune cells that are able to recognize cells that are infected, which I'll get into next week. But a cell doesn't know that it's actually getting infected when this whole process is happening. It's not until the immune system gets alerted that it ends up secreting these things called cytokines. And there's a shit ton of them, honestly. But the first round of cytokines basically tell other cells that there's an infection going on. Go ahead and protect yourselves. And I'll get into all of that later on, but the actual initial process of the virus entering into a cell, the cell has no idea what's going on. It just has these receptors. The spike proteins have an affinity or certain proteins that are able to really almost like a magnet. Think of it as affinity as a magnet to these receptors to go ahead and bind to it. But yeah, it isn't until the immune system gets alerted that other cells are aware that an infection is going on, and then there's going to be some things that happen. But to not confuse you, I will not get into that right now. Okay, so basically what's going to be happening is, so here I have an image of the spike proteins that are on the virus. And so after the binding of the ACE2 to the spike proteins, this protease is basically going to cleave the ACE2 binding domain on the virus. And what's going to end up happening is it's going to end up causing it to open up. And once it opens up, it ends up exposing those fusion proteins that I was talking about in the last slide. And so when those fusion proteins are exposed, it ends up having an affinity or acting as a magnet to be able to bind to the actual membrane of the host cell. And so this is basically the whole process of what ends up happening. Again, you don't need to know this whole process, but I just think these are very beautiful images of how these receptors are able to facilitate this whole process. Once the cell is, or once the virion or virus has fused with the membrane, then it's going to deposit its genetic material. Are the ACE2 receptors already on the host cells? or part of the virus? Good question. So the ACE2 receptors are going to be on the host cell. So it's really these proteins that I had mentioned earlier, the structural proteins that are within or on the surface of the virus. It's these, and I probably could have highlighted better. This right here is basically the host cell. So the host cell is going to have certain receptors that certain viruses are going to like to bind to. So I'm going to highlight, I forget in what slide, what cells or tissues express the ACE2 receptor, and this explains why there are certain people that are more susceptible to the virus and why prognosis is a lot worse for them. So here I have basically some key things you really want to know about the virus genome. So the virus genome, again, is a positive sense RNA virus. Compared to us, where we express over 20,000 genes, this virus, relatively small, it only expresses 10 genes. And unlike us, where we have those 20,000 genes that encode for over 100,000 proteins, these 10 genes only encode for approximately 27. And these proteins are basically a combination of structural, non-structural, and accessory proteins. And so again, the structural proteins are going to be what make they're going to be the sequence that encode or make up the envelope, membrane, spike, and nucleocapsid proteins. The non-structural proteins are going to be the sequence that encode for enzymes that are going to help with the whole replication process. And some of it's going to be involved in the actual facilitation of assembling the virus together, because the virus is made in pieces. And then the last set of proteins, which I won't highlight too much because I think scientists are still in the process of trying to figure out what these are. But within the genome, there's these accessory proteins. And basically these accessory proteins play a role in modifying the immune system or certain pathways to basically hide. And so if you ever end up, taking any kind of virology or bacteriology or anything related to that and learning about the immune response, you'll learn that there are pathogens in general like to hide from the immune system or manipulate the immune system in different ways. And coronavirus is no different, but we still really don't know what are some of those mechanisms. But basically they inhibit cells from being alerted that there's an infection going on. And if that ends up happening, then the virus is able to hide and replicate a little bit better. Somebody asked, is the RNA inside the nucleocapsid? Yes, that is correct. So the nucleocapsid is going to be, and all viruses have a nucleocapsid, and it's just going to be proteins that protect the whatever the viral genome is. So in this case, a positive sense RNA, from getting degraded when it's being assembled within the cytoplasm of a cell. So in order for the virus to even begin replicating, it needs to make replication protein. So this is going to be the first stage of after the whole viral entry begins and the genetic material is deposited, the host cell ribosomes are going to translate only a portion of the actual RNA strand. So here we have an RNA strand, and here we have a host ribosome. And so basically the sequence highlighted in green is going to be the non-structural region. And again, this non-structural region is going to encode for proteins that are meant to facilitate enzymes that play a role in viral replication. So it needs to make a couple of these proteins in order to even facilitate this process. And so depending on the different kinds of viruses, different proteins are going to get made. And so, and it also is dependent on how big the genome is. And so because this genome is relatively small compared to other viruses, to be efficient in making these proteins, basically what this virus does is it makes this giant protein when this host ribosome is translating the sequence. And then once the sequence is translated and there's this really long polypeptide chain, there's actually those proteases, or again, those enzymes that are able to break apart or degrade proteins. And so in this case, they end up just breaking apart the proteins, where they're able to cleave it or fragment it into smaller proteins so they can be assembled for whatever they need. And so this is how it's able to, even though it's only encoding 10 genes, this is one of the ways that it's able to make 27 different proteins, because there's a couple of genes within this non-structural region that can basically get fragmented and make up a bunch of different proteins. And again, these proteins are mainly going to be used to facilitate viral replication. And so with these particular proteins, the main ones that I really want to highlight is this replicase transcriptase complex. And so it's a complex, as you can see here. So it's made up of a bunch of different proteins that are needed for the RNA genome to get replicated. But the key enzyme that I really want you to understand or know from this whole complex is the RNA-dependent RNA polymerase. And I know it sounds complicated. I remember when I was taking virology in undergraduate, I was completely lost in what this meant. It wasn't until years later where I started doing viral work where I understood this. So it's not too complicated, just break it up. So we've learned about polymerases in lecture 1 or 2. And so polymerases are just going to add, nucleotides during the replication process. And so when you add on the RNA, it's basically going to be adding on RNA nucleotides, so AUGC. And what this RNA dependent refers to is this RNA polymerase needs an RNA template in order to facilitate this replication process. And so with this particular virus, what ends up happening is the positive sense strand ends up getting replicated and making a negative sense strand. And then from this negative sense strand, sorry about that. 1 second. All right. From this negative sense strand, it's going to end up duplicating and making a second positive sense RNA strand. Why the virus does this, I'm not 100% sure. But what's important to note is that this strand that it's synthesizing here is going to be used to during the assembly process when it's trying to put the virus all together. So remember, we have the structural proteins, and then we have the genome. And so it's going to replicate the genome, and during the assembly process, going to end up inserting it into the actual envelope and structure. So once this is replicated, the next step of this process is structural proteins need to be made. So again, these are going to be the ones that are going to encode for the membrane, the envelope, the spike proteins, etc. And so instead of the ribosome using or ribosomes being used to synthesize proteins from that positive sense strands, for whatever reason, and again, I don't know why these viruses do this, it basically ends up synthesizing a negative RNA sense strand, but only for the structural region of the genome. So here we have the non-structural segment, and here we have the structural segment. From this structural segment, it basically is going to end up facilitating another set of copying where it's going to synthesize the positive sense strand. And this positive sense strand, once it's done being synthesized, has a specific name because it has a specific function. And these new synthesized strands are called subgenomic messenger RNAs. And so these are going to be what's actually used by the host ribosomes to synthesize some of these key proteins. And these key proteins are also going to be used to help facilitate the structure, but also some of these accessory proteins that I was referring earlier to help hide from the immune system. So the way that the hijacking of the secretory pathway goes is it's all dependent on where these structural proteins are being synthesized. And so if we go back to lecture 1, when we were learning about the central dogma and how during the translation process, after the RNA is exported out of the nucleus, it goes from the endoplasmic reticulum to the Golgi apparatus, and then it gets secreted to wherever it goes. So the viruses are going to use this system to go ahead and get exported out of the cell. And then going back to the different kinds of where ribosomes live, there's 2 parts. There's the ribosomes that live on the endoplasmic reticulum, so the rough ER. And this is where the synthesizing of the S, E, and M viral proteins are going to occur at. And then the nucleocapsid, or the protein that is meant to protect the RNA strand, is going to be synthesized where the ribosomes that are found in the cytoplasm. Once all of these components are made, basically they need to be packaged together, and they end up getting packaged together in this intermediate location between the endoplasmic reticulum and the Golgi apparatus. And so this compartment doesn't have a really complicated name: endoplasmic reticulum, Golgi intermediate compartment. Makes sense. So this is the area where all this assembly is going to be occurring. Once the virion is assembled, the virus is going to undergo this process called exocytosis, or out of the cell. And so it's important to note that exocytosis isn't just a process that happens during viral replication or viral exit of a cell. It happens naturally when cells are trying to export things out of their cells. So if they're trying to signal to another cell or to itself, etc., it uses this process. But the viruses basically exploit this process so they can go ahead and get out of this cell. And once they do this, they basically can go ahead and repeat this whole cycle by infecting other cells that express the ACE2 receptor, and create a lot of problems for people. And so here I have a summary, 2 summary slides, basically going through this whole process. It's a very, I don't know how you guys are feeling about it, but I think it's a bit of a complicated process. So here, just for study purposes, just to understand like the different steps, I'm just going to go ahead and review it really quick, because I feel like the more times you hear this stuff, the better some of this might stick. So the coronavirus, again, is going to bind to the ACE2 receptors via their spike proteins. The protease TMPRSS2 will facilitate the cleavage of the binding domain on the ACE2 domain of spike protein. And this is going to allow the spike protein to open up so the fusion domain is exposed. The fusion domain is going to basically facilitate the viral entry by fusing with the host cell membrane. Once this fusion occurs, the positive sense RNA viral genome is going to get deposited into the host cell, and then the host cell ribosomes are going to translate the non-structural proteins of the genome. And again, these non-structural proteins are going to encode replication enzymes. The main replication enzyme, again, that you need to understand is the RNA-dependent Rna Polymerase, which is part of the Replicase transcriptase complex. And this complex is going to facilitate the replication process of the viral genome. The RNA-dependent Rna Polymerase will indirectly promote the synthesis of the viral structural proteins, and I went really in depth on that whole process with the slides, so go ahead and see the slides for those details. But it's not until certain sequences have been synthesized that host ribosomes are able to come in and basically synthesize those structural proteins. All these viral components are going to be assembled in the endoplasmic reticulum, Golgi intermediate compartments. And then once all of this is all assembled together, then the virion is going to undergo exocytosis, where it's going to leave the cell and infect other cells and repeat the cycle. So I think it's important to note that it's important to like mention or say that coronaviruses don't want to kill us. I know we've had a lot of problems in this last year, but in general, viruses don't want to kill us. And you have to understand or remember that viruses need a host cell to live. So they're an obligate intracellular parasite. They need to be inside a host cell in order to replicate. And if the host cell or host dies, the virus ends up dying with it. And so this explains why some of the most lethal viruses, such as Ebola, which has, I think, a mortality rate of like 90%, isn't able to lead to a full wide scale pandemic. And it's just because it kills so quickly. And I'm sure, you know, the way it spreads as well isn't very efficient compared to some of these respiratory viruses. What does kill us with this virus, unfortunately, is our immune system by this process called cytokine storm. And basically, it's just the immune system being overwhelmed with the amount of virus that's present. And it's producing a lot of these molecules called cytokines to try to kill off the virus. And in the process, it just goes in overdrive, and that's usually why patients will end up succumbing to the infection. And I will get more into depth when we actually are in the immunology classes next week. But it's important to note that it's not the virus itself, it's our response to the virus that's leading to death in these patients. And so it's also important to discuss why there's certain people that are more at risk than others. And to understand this, you have to understand a little bit more about the angiotensin converting enzyme receptor 2. So the ACE2 receptor that's needed for the facilitation of the infection of cells. And so the ACE2 receptor is expressed on specific host cell tissue. So it's not found everywhere within the body. It's found on cells that are in the heart, the liver, the kidney, lungs, blood vessels, and intestines. And so the reason why it's one of the symptoms for people getting diarrhea with COVID is because they end up, the virus ends up making it into their GI tract and ends up causing basically symptoms from your immune system responding to it. But the majority of the problems come from the organs that are implicated in the cardiovascular system or anything that's linked to the cardiovascular system. And that's going to be the heart, kidney, lungs, and blood vessels. And so within the cardiovascular system, the ACE2 receptor is important for regulating blood pressure and preventing tissue damage. So there's this whole process called the Rast system, and I'm not going to get into it because it's kind of complicated, and I think it's beyond the scope of this class. But basically, when your blood pressure gets too high, or if you end up getting thirsty or something like that, this system regulates all of that. So you have an equal amount that's not going to lead to high blood pressure and whatnot. Well, what ends up happening is if COVID's in using these receptors to get into the cell, that means that there's less of these receptors that are expressed on the actual cell. And so if this receptor regulates this molecule, angiotensin 2, and there's not enough of it, what's going to end up happening, it's going to increase the amounts of angiotensin 2, and this is going to lead to high blood pressure and tissue damage. And so the people that are most susceptible to infection are people that are type 2 diabetic, are hypertensive, or are suffer from cardiovascular disease. And if you have all three of these, the chances of you dying are quite high. And this is because of how all of this system is connected. And again, what kills the patient is inflammation. It's not the virus itself, it's our response to it. And so I'm going to highlight a lot of these diseases when we're talking about the macromolecules of life. But it's important to note that these diseases, or people that suffer from these diseases, tend to have higher amounts of inflammation that's going on in their body. And it's all due to diet. And so if you already have lots of inflammation going on in your body, and then you throw on a virus on top of that and your immune system responding to it, you're going to have a lot more inflammation that's occurring. And I'm sure you guys all know inflammation is not good. And so basically, again, highlighting some of these organs that are involved in the cardiovascular system, this regulates this whole system. And so if you end up having a lot of damage because of this infection, this is going to end up leading to the patients dying. And so before I end the lecture, I found this beautiful diagram where it highlights a couple of drugs that we're able to use to basically inhibit this viral replication process. I think because we just went over the whole process of how the virus goes about replicating and hijacking our host cell machinery, it's a good time to discuss some drugs that we have to go about preventing the virus from doing its job. And so the first one I would like to talk about is Camostatin Meselate. And so this is going to inhibit this protease that's involved in the whole cleavage process, so the virus is able to fuse to the host cell membrane. And so you're gonna learn that it's not just with this virus, but other viruses, all antivirals are targeting a specific area within this replication process. And so another drug that is important to know is EK1C4 HR2P. And so the name isn't so important. It's more of understanding the function of it. And basically it's going to inhibit the fusion of the virus to the host cell membrane. And then the other one that I'm sure you guys have heard about a lot in the news is this remdesivir. And so these are going to end up inhibiting this RNA-dependent RNA polymerase that facilitates the viral replication during this whole process. And so it's really important to stop this because you basically end up inhibiting downstream production of these proteins. There's a couple of other ones that you're feel free to look at, but during the lecture where I'm talking about the therapies that we have against the virus, I'm going to talk a little bit more about how these therapies actually work. So right now, it's just a good idea is to be exposed to what some of these drugs can do. And what area they inhibit within the actual replication process. But the actual mechanism of action, I'll go a little bit more in depth in a couple of weeks. This was a shorter lecture and I don't have any questions for you guys today. I will go ahead and put in the sign-in sheet into the chat. And if you guys have any questions, let me know. And if not, you guys are free to go. So